attempt.
Oh, Mike V, here you go.
Easy control.
I'm watching.
Oh my gosh. What is that?
Oh.
My gosh,
It's
that's
wow. I've never seen that.
It's an Asperoteuthis.
an Asperoteuthis.
Yeah, we saw one in the deep Gulf of Mexico 2 years ago.
Quick.
Okay. You have control of both cameras?
It's really interesting because we've been finding them for about the last 10 years in the Gulf and Caribbean, and before that they were only known from the Indo-West Pacific.
Really?
We don't know if that's because nobody looked here before or if they've just recently moved into the area.
Lining it down in normal position.
So have any been collected for identification?
Yeah, we've trawled some as part of the work that was where we were looking at the Deepwater Horizon oil spill.
Okay. But we haven't actually started the transect yet, correct?
Correct.
Okay. That was a bonus. That was pretty
That's
amazing.
Right. We'll get 10 minutes. That's a very unusual squid, and the species is Acanthoderma. It's Asperoteuthis acanthoderma.
Okay.
I'll type it into the event log.
Oh, great.
Coming to port.
And we're exactly at 800 meters, 800. Our temperature is 7.5 degrees. DO, dissolved oxygen, 4.29 milligrams per liter.
All right, found you on cam. You're looking good. Push forward.
Pushing forward.
Start bringing my heading.
That was a good catch by the pilot.
Yeah, thanks, pilot. Good job. Good work, pilot. Good work, video.
Thank you.
Now remove 7-8. 7-3.
Ooh. That's awesome. Mike V, the way he's moving his mantle, that's pretty interesting.
Fish eye.
Almost
Well,
like
most
spiraling.
of that movement you saw was actually fin.
Oh, was it the fins? Okay.
Yeah, that squid is pretty weird because it's got a long skinny mantle and then it's got a big fin, and then out past the fin it's got a tail with dots on the sides of it, so it's a little thing as to what you're looking at. But it was swimming by flapping its fins.
You're in camera.
Laterally.
Delta 15, looking great. Camera up.
Copy.
Did you guys miss it? Oh, we have. It's really awesome video. Some of the folks in the other room missed the squid, so they're coming. They came into the control van to see a replay.
Degrees.
Copy, 60 degrees.
That is amazing.
Definitely should be on the cruise highlights.
Definitely.
Along with the Dumbo from yesterday.
Yeah, I missed the Dumbo. I stepped out of the room, I think, for lunch. Or no, it was in the morning.
Looks like a cross current, or is that just from my motions on the tether?
It's still catching up with us.
Yeah.
Camera is 55. Delta 15.
Nav, Bridge, go ahead. Okay, where would you like to go? Okay, 800 meters, 073 at 2/10 of a knot. Is that
Well,
correct?
they're starting to also
Okay,
mention
copy that.
that
Dial it in.
when I've seen
Okay, there's in here while we
Asperoteuthis
wait.
in sub videos in the past, they've always been very
Yeah,
close
you're
to the
clear
bottom. So it was interesting to see this one so
Delta
far up the
15, my
bottom.
heading's finally 73.
Yeah, and we're over about 4,000 meters water depth right now.
Partial.
Try to get this little red guy here. You want to come in?
Somebody just join us?
Buenos días, Graciela here.
Buenos dias, Graciela. How are you today?
Goes from giant squid to smaller red.
Muy bien, gracias. ¿Y
Full
tú?
wide.
Full wide. Thank you.
Muy bien.
Qué bueno.
You want to,
I
just
just heard that I missed something nice. What did I miss?
An Asperoteuthis acanthoderma, this amazing squid. Did we get a size estimate on that squid? Did you get one, Mike V?
What's that?
I had science in my ear.
I didn't get a size, but I would guess it was probably
New caller on the line, which volume
maybe a meter long.
Wow. Nav, RV, will you just let me know when we start this transect? Okay, thanks. Okay.
Is that what they
thank you.
call the giant squid here in the Caribbean?
No, it's not a giant squid. It's a very strange squid.
Oh, there's something out there. Up, though.
So what was the name of it so that I can look it up, please?
Perhaps
The
a
genus
jelly.
name is
I
Asperoteuthis.
can see that. Yeah, that's correct.
Right.
Touching frame.
Yeah, hold the zoom.
If you go to a site called the Tree of Life website, they have a page on Asperoteuthis.
Yeah, video. If you could, stay
We're
full
starting,
wide
Nev.
and
Okay, back on shore side, we're starting the
hold
Midwater
the zoom
transect
until
now.
I request it, just so I can get relative motion.
Roger.
Let me know any information you'd like from me, pilot.
All right. This guy's coming in quick.
I have something coming into view here. Looks like a jelly or a larvation house.
All right, come on in, video.
Coming in. Aye. Holding.
Really long tentacles.
All right, you can come in tighter.
Plenty of time.
That's a jelly. I'll let Mike Ford
Wow.
start talking.
All right. Josh.
Got dark.
It's great. This looks like a little track in the deuce, it seems to be.
There you go, video.
At
Very
quick
nice.
glance. Really nice long tentacles there,
Watch my
and
hand.
you can see
Yeah,
below
you're fine now. Don't
the
use
bottom
too
of
much
the
on it.
umbrella, there's a little shelf of tissue kind of
Got
extended
another
downward.
frame.
That's
Yeah, you're gonna come out
kind
here.
of
Can
a
let 'em
velum.
Yeah.
The
Coming out
narrow
nice.
the aperture at the bottom of the umbrella to help this
Wide.
jelly swim a lot faster.
And is that one of the defining characteristics of this group, the Trachymedusa?
That and the radial canals and the placement of the tentacles along the umbrella.
Okay.
Your camera's pointed up just well, I take that back. Value just changed. Carl, did
And
you
do
bring
they
the lower
also
sternum gel?
hold? Do they also brood their offspring
Yeah.
Yeah,
within the bell?
yeah.
Would you like me to bring
Yeah,
that?
actually,
Yeah, I brought 'em out 'cause it gets rid of
interesting
some of
ones
the-
that we saw yesterday.
layer.
Chrysota,
Especially the
is
starboard
known
one.
for
Will do.
developing
But I
right from
tilted
the
them
ova,
out and kept them
small
both out.
medusae
Yep, you got
that they keep up in the bell.
Push
That's,
it forward a
that's
little.
Delta
fairly
18.
common among the jellyfish to do
Hunting
that.
into the
Okay.
darkness.
Thanks, Mike.
Something lower right.
Yeah, it looks like maybe a gelatinous.
Not sure.
Something lower right coming up, possibly. Oh, yep. Wow, good eyes.
All right, come on in, video.
Pushing. Still pushing.
Yep.
Still pushing.
All right, hold there.
Holding here. There he is.
All right, come on in. He's
Coming
drifting
in.
to my port.
Concur.
Yeah, he's way out there.
There's some gain here.
Is that a
That
mucus
looks
house?
like the filter structure for the
Looks
house
like he's
of a
in a mucus
larvacean.
net of some sort. Mucus
Hey,
ball.
come on out, video. I gotta
Coming
readjust.
out.
A larvacean
Right.
is an invertebrate,
So it looks like
but it's
everything's
related
drifting to
to
my port.
the vertebrates.
So I'm gonna have to.
And the
set up
animal
on my
actually looks
starboard
like a
and
fish
try to grip
larva
all of it.
inside of there, and it'll be inside there beating its tail, setting up a current that causes water to flow through the filter, and it traps very small particles that the animal eats.
That was a nice image too. You could really see the bigger extent of the mucus kind of surrounding the house.
Right. The main thing that you can see there was the inner filter structure, but there's a very large, almost completely transparent outer mucus structure as
Motion arms are
well.
deployed.
That's what
Yeah,
we're
it's
talking
Just
about.
slightly.
It's much better. Thank you, co-pilot.
Yeah, no problem.
So
Every once in
yesterday,
a while, the animal will abandon that whole structure, swim off, and make a new one. And the structure will actually sink down to the bottom and carry all kinds of detritus and marine snow with it. So
HPU
it
secure.
transports carbon to the ocean
Heading
floor.
still 73.
so that's a pelagic
Camera
swimming tunicate,
55.
correct?
Delta 16.
That's right.
And we observed benthic tunicate yesterday. If anybody was watching the predatory tunicates with the big hood, we were looking at that animal. It had the potentially symbiotic polychaete associated with
Oh,
it,
here
so
we
that's a
go.
benthic tunicate, and we just saw a
Coming at us
relative
really
swimming
fast.
pelagic tunicate. And we have something here on the bottom right.
I think he's inside our lights.
Yep. Trying to get him.
Shrimp, maybe?
He's coming
Yeah,
at
I'd
me.
say shrimp.
He didn't want to cooperate.
No close up on that one.
Another thing coming in that's moving towards your port. You can see it looks like another jelly of some sort. Just drifting down almost to the center frame now.
Yep, tracking. May come on in video.
Coming in now.
Another mucus house.
Yeah, this one looks like it might have been abandoned. I think we've got
Let this
enough
guy
of that.
go.
Yeah, that's good,
Roger.
pilot. Thank you.
Lead
Full wide.
frame and coming out. Popping out. Full wide.
Can you give me an update on the depth that you're at and the plan for the Midwater transect?
Yeah, currently we're at a depth of 800 meters, 800. We're conducting 10-minute transects at every 100 meters until 1,200 meters.
Come
So
on in video.
Coming
we'll
in.
do 5 midwater transects, each for
Is that
10 minutes.
Really far out.
No, just really small. You
I
can see
think
it
it's
in,
very small.
see it in,
Is that a siphonophore?
Put my levels
Yes,
down
it
a
is.
bit
It's
here.
a siphonophore.
3 meters out.
Siphonophores
The
are
group
really...
that's called Physonex. They have a little tiny gas float right up
3.5
at the
meters
tip.
away.
And Mike, tell us a little bit about how the colony is set and how the different cells may function.
4 meters away.
can jump in. It's a really great organism. Siphonophores are sort of really great an example of the medusan form and the polyp form of hydromedusae being adapted and changed so that they have different roles. In this animal up at the top, most of those are nectophores, which are medusae forms that are really just responsible for propulsion now. And the lower half of the organism, where it's dense and white, just at the very top of the screen,
and
that has organisms that have been adapted for protection. Very close to the nectophores at the top are structures called dactylozoids, which are studded with nematocysts, stinging cells.
Thank you, video.
And then below, you'll see clusters of modified forms, one for feeding, one for reproduction, and some other ones for digestion. So working together, all attached to a common stem, the siphonophore can handle all of the functions and be rather large in the ocean environment. They're very popular all around the coast of the United States and all over the ocean, and are a pretty serious predator out here.
Thank you, Mike.
55 camera angle.
And there was just a fish on there, but it took off before we could get a close look at it.
Okay.
Sorry, I thought I heard you say we're up.
What
go
do
ahead,
they
Nev.
prey on?
Oh,
Do they
hold on,
prey on
Graciela.
fish larvae and eggs and things like that?
Hold on, Graciela. Go ahead, Nev. Okay, so that ends our 800-meter transect, and we'll head on down to 900 meters for the next one.
Timing lines.
Okay, go ahead, Graciela.
Watch is coming in a few minutes.
I was just asking, what do they prey on?
Let me ask.
They're said to be serious predators in the water column.
My guess would be large crustaceans and fishes. Anything else, Mike Ford?
Hold position.
Pretty much whatever they want. They can do small fish, tiny fish, great with zooplankton prey. They make really, really good use of those stinging cells. When you didn't see it on this particular animal, but when the tentacles that are attached to the feeding cells are relaxed, they can release out to be a pretty big branched net of tentacles studded with stinging cells. They can catch larval fish, certainly small fish, and crustacean prey.
Okay, we're just doing a quick pilot change.
Can I use this opportunity to communicate in Spanish?
Wonderful. Thank you, Graciela.
Sí, buenos días a todos. El barco Okeanos Explorer con Andrea y Mike y los pilotos de los ROVs D2 y Sirius están haciendo una serie de transectos en la columna de agua a una profundidad de 800 metros. Ese es el que acaban de terminar. Ahora van a bajar a 900 metros y así sucesivamente hasta que lleguen a 1200 metros de profundidad. Cada transecto lo hacen por 10 minutos. Si hay una serie de científicos comunicándose con el barco porque están tomando la información de los organismos que se encuentran en la columna de agua. Mayormente se van a ver medusas o aguavivas, se van a ver una serie de organismos en su mayoría gelatinosos, se van a ver tenóforos, se van a ver peces, así que están haciendo estos transectos para poder identificar las especies que están en la columna de agua y alguna relación que tengan. Una de las que mencionaron fue los larváceos. Los larváceos, aunque son invertebrados, están más asociados a los vertebrados porque es una de las medusas que parece una medusa, una agua viva, pero que tiene adentro un organismo que parece una larva de pez. Así es que estos cambian su estructura de gelatinosa de vez en cuando y algunas de estas se han visto sin el organismo dentro. Así que ha estado súper interesante. Aparentemente vieron un calamar no es el calamar gigante, pero son calamares bastante grandes. Yo no tuve la oportunidad de verlo, pero sí me estaban diciendo que es una especie que en español se pronunciaría
Matt, just to make
Asperoteuthis.
sure we have yet to do the 800
Por lo menos esa es la
This is the
pronunciación
beginning
que yo
of
le
it.
daría según lo tengo escrito aquí. Ellos van a estar haciendo esto cada 10 minutos a 900 metros, 10 minutos a 1000, 10 minutos a 1200, 1100 y 10 minutos a 1200. Después de eso entonces bajarán a la profundidad establecida, que en la próxima intervención les digo cuánto es. Así que estamos los científicos conectados a través de teleconferencia con el barco. La persona principal es Andrea, que es la que oyen que está describiendo la mayor parte de las cosas que pasan, y los otros científicos son los que intervienen y nos hablan sobre cada uno de los organismos que se ven. Así que espero que disfruten la buceada de hoy, y cuando pueda yo también me estaré comunicando sobre lo que está pasando en la expedición. Okay, that's it.
And video while we're doing this, if you want to look in the water column, that'd be helpful while we do this little dance here.
Yeah, sounds good. We're just keeping
Graciela,
an eye out
do you
for
have
any
an
more
idea how many people are
interesting
in Puerto
creatures
Rico
that
are
happen to
watching
wander through our
this
field of
feed?
vision.
You know a lot of people
Yeah,
that are
copy
keeping
that.
track of the dives and.
Well, from what I hear, a lot of people have, through Facebook, found out about what's going on, so they should be logging in. Of course, most people are working and in school. I was about to send a text to find out how many schools or how many kids were actually watching, because they apparently the science teachers and at school, they connect the monitors so that the kids can watch the expedition. So let me see if I can do some kind of estimation. In fact, you should see, I hope that I uploaded, or a friend of mine uploaded, the caricature that came out in the newspaper this morning about the Okeanos exploring the waters around Puerto Rico and the Virgin Islands.
Oh, awesome, great. Yeah, I
So
mean,
if
you
I
don't have
haven't
to,
posted it yet, I'll upload it.
oh, that would be great. You don't have to give an estimation. I was just curious if you had a general sense of how it's being, how many people are watching. That's all. That's great. I'm glad that students are tuning in and teachers are utilizing this. It's awesome.
No, but I would be interested in finding out, do you have a way of tracking the IPs that connect to, well, YouTube has the number of people that view the video on YouTube. Is there any other way of tracking the IPs?
I think they do. Casey Cantwell would know that, and Emily Crum. I think they try to keep a number and estimate a number of visits to the live feeds and to the web blogs and things, but I'm not sure if they can track from what locations people are tuning in. I don't know if they do track that, I should say.
And I'm going to find out if the Department of Marine Sciences today has the live feed, so let me go and find that
Okay.
for you.
Thanks. Great talking to you.
Shove ahead there.
We're crossing a depth of 850. 850.
Sorry. Pushing out.
Put a little right crab in there. Actually, I'm getting pulled a little bit, but there we go. That's happy.
Okay, so there should be the Department of Marine Sciences is hosting the watching the Okeanos Explorer between 9 and 11 this morning and between 1 and 3 p.m. This afternoon.
Wonderful.
So we should be getting a number on how many students stop by, and this is in the main campus, so there should be a number of people coming in from the other, physics.
On a related topic, I'm going to be out in the Sant Ocean Hall at the National Museum of Natural History at the Smithsonian Institution at 1 o'clock today with a large screen feed from the Okeanos.
Oh, great.
Talking to the public there about what we're seeing.
Depth at 875.
This is definitely a wonderful outreach tool. And we're about 24 miles northwest of Puerto Rico. We're about 12 miles east of where we dove yesterday. The bottom water depth here is around 4,000 meters, and after we finish these midwater transects, we'll head down to about 4,000 meters depth to survey the east wall.
Nav, how many transects are we planning? Okay. Copy that.
There are a number of fishermen that have been watching very what you're doing because they're very much interested in what the areas look like. Most everyone from the north, northwest, and west coast of Puerto Rico are really intrigued, although I heard also from some fisheries from the east and south coast who are also watching.
Depth 892. I'm going to slow at 895 here. Slowing down slightly. Pushing out. Coming back to center. Okay, that's 900. Copy that.
And we've reached our second target depth of 900 meters, 900.
So Chris, I'm going to run with less delta and bring my camera tilt up so you've got a little bit more
Copy that. I'll push
time
out.
there.
Roger. Pushing out.
And I will rise over the top of you if you come across something interesting as I go by. There we go.
It's going to be a constant crabbing battle.
Oh, that's all right. Just, do what it takes to make it work.
Yeah. You can kind of see that across the screen with the sea snow.
Nav, this is Washley. Just let me know when you start this transect. You started? Okay, thank you.
Okay, looks like I'm out there a ways.
That was the start, right?
Yes, start. So we're at the start of our second midwater transect at 900 meters, 900.
That's a little pool.
There's actually very little marine snow in the water column here. It's kind of surprising that there's so little particulate matter visible.
Okay.
And Mike V, compared to some of the last cruises that we've conducted midwater transects, there was quite a bit of marine snow in the canyons environments, correct? Off of the northeast U.S. at these same depths?
Yeah, there was a lot more marine snow there. And also, I think there was a lot more gelatinous organisms there.
Yeah, I remember a few times we came across the
Salp.
salps. Yeah, we were calling them salp parties at some depths. Just the whole water column was just covered with them. They were really dense.
Is there an expectation of there being latitudinal differences or seasonality to what you see in the water column?
There's definitely seasonal differences, and there are different geographic differences that are related to surface productivity.
Is there something coming up here?
Want to go snap zoom video? Just hold there.
That's another siphonophore.
Keep coming.
Looks very similar to one we saw yesterday.
Sorry.
Definitely different from the last one we saw today.
It's swimming very fast, too. You would think something like this would be a fast swimmer, but it's moving right along.
Yeah, he's going down pretty quickly. Might have to leave him here.
Okay. That's good. Is that good for you guys?
Yeah, we've got enough.
Okay, thanks.
Okay.
You can come out.
Come wide video. Let's get back up to 900.
Well, you asked about viewers, and I just got a whole bunch of email messages and
Did you?
texts saying of people who are watching. And Carrie Coos has screens ongoing all day long at the Department of Marine Sciences in La Parrillera. So there are people coming in and out all day to watch.
That's wonderful. And that was, did you say the Department of Marine Sciences?
Yes.
Great.
And also the University of Puerto Rico Rio Piedras campus has a ongoing presence.
Tug.
Yeah.
So formation flying is a little bit more tasking, isn't it?
And we're just over halfway in for our second midwater transect at 900 meters.
It's different.
Something coming up?
Where?
The spec?
Center
red
screen?
Spec?
Go ahead, video.
Sometimes it's better to just drive up on those tiny ones.
No, I was looking up. That's cool.
Is that full? There we go. He's small.
Yeah.
Come on.
It's hard to see.
So there was something higher up, more diffuse.
Any ideas on that, Mike V?
It's gone.
I don't know. It looked like it had appendages like a station.
Let's come back to this video.
An ostracod,
Hard
possibly?
to tell.
Okay, come on.
doesn't look like it's swimming. So I've mentioned marine snow before. That there's basically two major components to the marine snow. One is mucus, and the other is the exoskeletons of planktonic crustaceans.
Towards them too.
That may have been a molt from an amphipod.
Okay.
But that's just a wild guess on my part.
Here's something else coming up, another siphonophore.
Yeah. Moving down like the other one. Start to come in video. Hold there.
Another siphonophore like the other one. Most of what you see in midwater are gelatinous organisms. And if
this one, you can see the nectophores on the end pulsing a bit.
Yeah, it's swimming like crazy, but I don't know how it knows that we're here.
Has it got photoreceptors of any kind?
I'm not aware of any photoreceptors on them.
Might have to let him go, Jared.
Yep. We've got quite an acoustic and pressure signature as well from the props.
Thanks, pilot.
Is that full wide?
Oh, that was great.
Full wide?
Copy,
All
thanks.
cameras.
Thank you. Let's get back out there.
Fair to say, I mentioned before that most of the living space on Earth is in this deep midwater environment. Most of the animals that you see here, other than microbes, are these gelatinous organisms. So if somebody from another planet was coming to learn what lives on Earth, what they would conclude is that most of the life on Earth is jellies.
That's good with me.
Okay. Paying out slow.
Do a little auto heading test here. Hold it around 60 down.
Okay.
That was happy last time. If you were at 23,
22, 23, yeah. I just fly the tether slack.
Yeah, understood.
Are we going down?
We're going down.
We are.
Oh, I guess this ends our midwater transect number 2.
Yeah, may not have had you on this talk circuit there.
Sorry about that, watch lead. So
I had a request.
watch lead, can you not hear EXRV?
For the
We have to have a
scientists
separate
on
channel, is that right?
Telecom, by themselves, I probably have done it already, but people who tune in were wondering and they're interested in these comments about the majority of the Earth being water, and for that, as a fact, it's mostly that water column, and the resulting conclusion from someone observing the Earth that it would be mostly jelly forms. So I just got a message, asking who that scientist was, because it was very interesting comment.
That's Michael Vecchione with the NOAA National Marine Fisheries Systematics Lab. Sorry, National Marine Systematics Lab.
Lateral and auto head works pretty well.
We just saw an eel go by just briefly.
An eel?
Yeah.
Like a snipe eel? I missed it.
No, it wasn't a snipe eel. It was one of the shiny-sided things that look like cutlass fish. my mind just drew a blank.
Paralipetid, Barracuda, the ones that swim vertically.
Yeah.
Push out a little.
Maybe.
Yeah, Cerebelmer might be. That's what Randy Singer just typed on the chat log.
Oh, okay. I actually started looking up whether siphonophores have photoreceptors.
I would think our propellers have a pretty significant low frequency signature.
We're at a depth of 967 meters.
D2 passing 975 descending.
Just went past another siphonophore.
Same species?
It was pretty distant. I couldn't tell for sure. I mentioned yesterday that there are siphonophores that get over 100 feet long. So they are longer than a blue whale.
That's pretty amazing. And then to be able to communicate across that 100-meter length, because all of the cell types have different functions.
Depth 995, slowing.
Adjusting my tilt.
Copy. Pushing out.
So since we're just getting set up for the next transact, I'll continue telling stories. I was on a Johnson Sealing cruise one time when the sub had come back up and we were taking the samples off of the sub, and a guy got a siphonophore tentacle wrapped around his hand, and he jumped down off of the top of the sub and ran into the lab and tried to get it off of him because he was getting stung really badly. And the next day, his hand was swollen up to twice its normal size.
Wow.
And it had puncture marks in it that looked like he'd put it through a sewing machine.
Really?
They have very powerful nematocysts or stinging cells.
Coming out of autohead.
Are we ready to begin transect number 3?
Thank you, Nav. Yep. So we're beginning transect 3 now. We're at a depth of 1,000 meters, 1000.
We dropping any targets on these for location?
No longer fighting lateral, so it's interesting.
That's good. So find us a giant squid.
I'm trying, man. See if we can find something. Did anybody look in sonar after the last episode with the squid? See if you could actually see him as he left?
Don't know.
That'd be interesting. So if that was the case, we could...
To add to Mike's comment on the stinging cells. The stinging cells in most jellyfish, the medusae and the siphonophores that we're seeing today, are kind of nature's great solution to having a gelatinous body without enough structure to have a mouth to capture prey and hold prey. So these tiny cells have small harpoon-like structures, aggressive-looking barbs, and filaments that, when they're triggered by a physical stimulus or a chemical stimulus, will shoot out of this sac-like cell at thousands of Gs and penetrate a prey's body. And many of them will pump a number of different toxins into their prey to stop the prey from moving around. So nematocysts are common all over the deep sea and a great solution for a very lightweight gelatinous body.
Thanks. That was Michael Ford.
One of the great things about using a sub to actually look at what these animals look like is when we've studied them using historical methods like towing nets and collecting them that way, as soon as the net hits them, they fall apart. And one of those siphonophores, for instance, when it gets into a plankton net or a trawl net, would just break into a bunch of jelly-like pieces, and very, very difficult to tell what it is that you caught. Whereas when we see them with the video, we can see what they actually look like. Another advantage to using a visual observation like this is that we know exactly what depth that the animal is at. If we're towing a net, we might know approximately where the net was when the animal was collected. We don't even know that much, but some kinds of nets we can tell approximately how deep it was. But with a sub, you know exactly how deep it was and what the environmental conditions were where you ran into it.
And there's some nets that you can deploy closed and then trigger them to open at a certain depth range and close them again and bring them back to the surface, but they don't always work as you'd planned. So.
Well, that's true, and even when they do work right, they sample a layer that may be a few hundred meters thick.
Right.
Feel a little warmer in here today than
it has been.
It's a little warmer.
Comfortable in the control room for the first cruise.
There's something on the left.
It's usually a little on the chilly side in
it
here.
is.
So it's
Sky video.
pretty pleasant.
Aero Worm. Looks like it.
It is an Aero Worm, a ketognaph. It looks like a fish larva, but it's an example of convergent evolution, the very efficient body form for swimming. This is a very strange group of invertebrates. And they're also vicious predators. They have hooks at their mouth, and they grab copepods and fish larvae and things like that and eat them.
Life will get better.
We lose light though then.
Yeah, if you push him inside the light pool for sure, but unfortunately, without stereo vision.
So you mentioned
we don't have
convergent
range.
evolution, just to give another example to folks that may be a little more familiar with
Go ahead, video.
animals back on land. Another example of convergent evolution would be, say, the wings of bats and the wings of birds. So both of the wings are used to fly, but not necessarily made from the same anatomical structures.
What do you think the size is on this, Mike?
Lasers coming on.
The deep ones are larger than the ones up in the surface water, so this one might be a few centimeters long. I'm just guessing off the top of my head.
Come on, video. Something to my
Educated guessing.
starboard, Dave. I don't know what that would be, but I can get eyes on it quickly.
Oh, that gelatinous tilting down.
I don't know what happened to it.
I think you blew right past it.
I just saw on the chat log that Peter Weeby is at the Hui thing, and he'd be better able to answer that question than I would.
I don't know where it went. I think it's still in between you and I.
Yeah.
Yeah, that thing you're looking at is too small.
Can't get there from here.
It's not too small. It's just in between us.
Just out of our...
Yeah. Push back out, back down to 1,000.
tilting out. Angle set. 30 degrees.
Copy that.
So are we done with Midwater Transect 3?
One more minute.
One more minute, okay.
And that wiggly thing might have been a fish, but there are other things that might have been too.
And that concludes transect number 3 at 1,000 meters.
Thank you, Nev.
We must.
Mike Ford and Mike V, I was watching some of the Midwater Transects yesterday, but not paying attention as much as I am today. Do you see what you saw yesterday versus today, a difference in, where these midwater fauna were most abundant at a certain depth range, or any comparisons to make so far?
Well, the depths that we're sampling is actually a fairly narrow depth range. And, it's going from the deep mesopelagic, or what some people refer to as the twilight zone, and into the upper bathypelagic. And as you get deeper in the water column, in general, you see less animals. But it's not entirely true. You can run across very narrow depth bands where things are concentrated, and you might encounter much higher abundances within just a layer just a few meters thick. But people that have been watching these as we're doing them will probably notice that we see more things at the 800-meter depth than we do at the 1,200-meter depth, usually.
And we're
I mentioned yesterday, and I don't think I mentioned today yet, that one reason that we selected this depth band is because this is also the depth at which deep diving toothed whales feed. So things like sperm whales and beaked whales will feed in these depths.
I have a question for you. We often come across surveying the bottom these kind of areas on the seafloor that are dug out, and a lot of people often say that's from a sperm whale. Has anybody actually observed that happening where these sperm whales dive down into the sediment and create these marks on the seafloor?
There's an example of damage to a submarine cable where when they hauled it up, there was a dead sperm whale that had tangled up in the cable. And they think that it got its jaw in the sediment and got underneath the cable and got tangled up in it.
Okay. Thank you.
But most of the speculation about those pit marks being from deep diving whales is a result of a paper that was published based on observations in the Mediterranean, where they measured some of the pits. I think it was on a seamount, and they calculated that the measurements were consistent with the lower jaw on a beaked whale.
Okay.
Go in more. He's kind of at my speed.
Trying to keep up with you.
So we got a fish on the screen.
We sure do. Looks like a paralipetid, but maybe a larval type.
He's moving 15 to 20
I think a
meters.
paralipetid is a good guess.
Yeah, I think they often have those black blotches along the stomach area.
Can I keep coming, video? Yeah, hold there. It's a little tough, sorry.
Yeah, he's not cooperating very well.
Yeah. I think it's likely it was a paralipetid. When I first started in marine science, my master's degree was focused on larval fishes off of North Carolina, and I ended up sorting plankton samples for larvae, fish larvae, counting 20,000 larvae, and identifying numerous species. So it was a good introduction to ichthyology for me. You know, you can go out with a net and collect lots of different fishes from different families, different orders, and I learned a lot from anguilliforms, the leptocephali eels, all the way to the puffer fishes and flatfishes.
Thanks. Might still be there.
Just tried to see a shrimp.
Go ahead. He's coming down too.
I see him.
Looks like it's on skis almost with the antennae. Oh, there.
I wonder if they're following
a
light.
A suggested kind of shrimp.
1082 meters.
And the suggested
They have those
can
long antennae that are trailing backwards, and they function sort of like the lateral lines on a fish, except they're farther out from the body so that they can detect things farther away.
So it's a good defense to evade predators. They also are able to spew bioluminescent ink, so to speak. Correct? The Sergestes? I know some of the midwater shrimps can do that.
Yeah, there are some midwater shrimp that can spew bioluminescence.
It's not quite as fast as the other guy.
He's not running away from us, though.
Another thing this is a good example of is the red color that you see at these depths.
At least he wasn't.
The reason for that is that there's no red light down there. Red light is absorbed very quickly in seawater. And if there's any light at all, it's blue. And the blue doesn't reflect off of the red of the animals, so that it makes the animals effectively disappear.
Except for there are a few fishes that have evolved the ability to produce red light. I believe the malacostiids. I think Randy Singer commented on that yesterday as well. So if a fish can produce red light, then they're able to see animals that are red.
Yeah, the one is called a stoplight loose jaw.
Yes.
And the reason it's called a stoplight is because it's got little red lights underneath its eyes. That's a good way for it to see the red shrimps.
Okay, coming up on 1100. That's it.
Okay, we're coming up on 1,100 meters. Let me know when we start the transect, please.
We can start it immediately, can't we?
Oh, okay. I always thought you guys had to get a little settled first.
You settled, pilot?
Settled.
Settled, ready to go. Thank you.
Bringing my tilt.
Chris needs no prep.
First what?
Chris needs no prep.
It's debatable.
Debatable. All right, we're in the start of the 1,100 meter transect. Time is 10:42 local time, UTC 14:42. And this is the 5th dive of the Oceano Profundo expedition. Conducting midwater transects before we go to a bottom depth of about 4,000 meters. I hear a clicking in my ear. Can we... Is that from shoreside or from us?
I'm hearing
Video?
it too, so I think it might be a
Video,
short
can you come in on this swimming creature here? See if we can at least make an ID. He is tiny.
It's like another fish. Guessing another barracuda.
Of my tether will reach out that far, but
You don't have much to work with there. But I'll catch up.
nice work, video.
It's kind of hard to see. It's got very large pectoral fins.
He's swimming opposite direction of all the others.
Yeah, that one looks kind of unusual.
Yeah, I'm not sure that was a paralipetid.
So you might be able to get him again unless he's...
Did it look like it had photophores along the belly there? Along the ventral side?
Maybe we can get a good frame grab.
Get this guy while we're here.
Zooming in on another crustacean.
Go ahead, video.
And then there's an arrow worm to the right.
Yes, another suggested shrimp.
Okay, come on. I think he came at me. Is that full wet?
Full wet.
Thanks. I'm gonna get this Aero Worm over here. Seems to be in the middle of the light there.
You know, getting back to your earlier question, we're seeing stuff today that we didn't see yesterday.
Yeah.
I don't know if that's because we're just adding to the volume that we searched, or if there's really differences along those kind of spatial scales.
Yeah, I'm interested to see when we get to the bottom today as well. We're going to be in the same depth range as we were yesterday, and it looks like the geology may be a little bit different. So we may be crossing over more hard bottom today and less sediment. So be curious if we see a lot of different organisms at the bottom. Is that right? soft segment.
The only thing about staying way out in front, Dave, is we have more time if things are stationary, but if we see something on the periphery and they scoot away, we don't hardly get any glance at it. But it's
generally,
trade-off.
if something can scoot, chasing it is frustrating.
Yeah.
It's better to creep up slowly.
And we're conducting our 1100 meter transect, 1100 meters.
Get a snap zoom on this item.
Is that a salp chain or a siphonophore?
I think it's a baby siphonophore.
It has no color.
Stand by one. Come a little wide video.
What causes the red color in the siphonophores?
Ready to go.
Tug.
Sorry. Just
puppying off.
There's so much out there to see, Dave. Just gotta go find it.
All good things come to those who wait.
We just got a email from Graciela of a cartoon that was in a newspaper of the Okeanos Explorer. It's very cool. From the media.
Is it potential T-shirt art?
Thank you for sending that.
Yeah, I was in the newspaper this morning.
Oh,
so I hope they'll keep a couple of the original ones.
Come on, Squid.
That's awesome. I can't sit still today. I've been standing up and sitting down every couple minutes. I think this needs to be raised a little higher so you can see over.
You've got front row envy.
Yeah.
Maybe we can get a better monitor for back row.
This has been discussed.
We could do like stadium style.
Yeah. Movie theater style. Okay, we're still at a depth of 1,100 meters.
We got 2.5 minutes to go on this transect.
Is your aft thruster misbehaving, Dave? Sure seems like it.
It says 0, but I don't have a status.
It could be a speed or current.
Well, they should
Fault.
operate in tandem. Let's see what we've got here.
Yeah, over
Sure enough. Good eye. Staring at the video is
What is
so
it?
alluring. Oh, we're having a technical discussion here.
Oh, okay.
Sorry.
Called attention to my lack of attention. But
Not intentionally.
my attention was elsewhere. It wasn't lacking. It was just
What speed are we doing?
0.2 knots.
Let's try to do some macro here. Video start to come in. Any day.
It's like
It's out of the lake pool.
Is it close?
Yes.
Okay.
That's full wide.
Copy. Yeah,
thinking that it's another Aero Worm.
Yeah, I think it is a Keating now.
Okay, come back in. Okay, come on. Thank you.
See how fish-like it looks when it swims?
I'm starting to realize the light pool is different than normal. The swing arms askew.
And you mentioned earlier,
Come back in,
Mike V, about Convergent Evolution, and just to explain to our viewers a little more about what that is, it's really evolving characters that are similar because of similar environments. So, for example, this resembles a fish larvae, this Keating nath. They both live in this open water pelagic environment. Similar, as I said before, to the wings of birds and the wings of bats. They both have the ability to fly, but the structures that allow them to do so aren't necessarily from the same origin.
Right, that is the end of our fourth transect. We can descend another 100 meters when ready.
There you go. Setting my tilt to 45. 45 degrees, paying out slow.
Copy that, coming out slow.
Just build up the speed as you get position.
Pilot, I was promised a squid. Came on.
You missed the
I know.
first one. That was pretty awesome.
And then Mike says that there's more things at 800 meters than 1,200 meters. It's just...
So we're moving to our 1,200 meters now.
We are descending.
Okay, we have one more midwater transect.
Andrea,
Can I switch to Spanish now?
Hold on just one second. I have a question from the pilot. Go ahead.
She can go ahead. I was just going to ask why there are more things at 800 rather than 1,200. Is that just closer to surface related, or?
So I'll let Mike V answer that since he's been talking about this area for a while. Mike V, do you want to answer the pilot's question?
Yeah, for the people that didn't hear it, he was asking why there's more stuff at 800 meters than there is at 1,200 meters, and he basically answered his own question. It's because that's closer to the surface, which is where the productivity occurs. All of the productivity in the ocean, except very close to hydrothermal vents, all of the productivity occurs in the surface layer, the upper 200 meters of the ocean. So as you get farther and farther away from that, there's less and less food available, basically.
Great, thank you, Mike. And Graciela, go ahead. You can do a translation for us.
Okay. Sí, el Okeanos Explorer está ahora mismo a 24 millas al noroeste de Puerto Rico y están en el último transecto que va a ser a 1.200 metros de profundidad, un transecto de la columna de agua. Mayormente lo que se ha visto hasta ahora han sido medusas y la mayor parte de los organismos gelatinosos, y han estado hablando los científicos que están en diferentes sitios de Estados Unidos y conectados al barco a través de teleconferencia. Así como soy yo de San Juan, me estoy comunicando con ellos en el barco. Y han estado hablando sobre los organismos que se han encontrado y que mayormente han visto mayor cantidad de ellos en las profundidades de 800 metros o así, porque la productividad primaria está mayormente en la superficie y por lo tanto a 800 metros van a encontrar más organismos que en aguas más profundas. Sin embargo, sí hicieron un comentario de que habían encontrado menos lo que se llama marine snow, menos nieve marina, Me imagino que alguien me va a mandar un texto diciéndome exactamente cuál es el término que se usa en español. Y que hay una diferencia en términos de la estacionalidad de la misma y que puede haber más durante cierta época del año que en otra época. Si han estado hablando sobre los peces... are you going to describe that? I'll be quiet.
So we have a siphonophore here. Mike Ford, you want to describe it a little bit for our audience?
Yeah, that's great. Beautiful siphonophore. You see a lot of the trailing tentacles at the bottom. And you can see a dense set of modified cells there, tentacles. A lot of them branched with many stinging cells. The modified cells on that end also include reproduction and digestion up at the top. Those modified Medusan bells are for propulsion. So it's a beautiful specimen. It's really big and kind of trucking along.
That's great, guys. Thanks. We can continue down.
Copy that. Thank you, Dave. Thank you, video. Full wide?
Full wide.
Y como verán, los pilotos que están manejando los ROVs, especialmente el D2, están muy pendientes a todo lo que está ocurriendo en el agua y así pueden enfocar específicamente en los organismos que se ven para que los científicos puedan captar la imagen y describirlos en el momento. Así que esto es una acción extrema entre la tripulación del barco, el capitán, el primero de a bordo, entre los pilotos de los robots, el navegante que está continuamente dando instrucciones para que el barco se mantenga en posición para que los robots puedan seguir obteniendo video del área alrededor de donde están. Hay dos robots, el de arriba, si van a la cámara número 2 de lo que se está tratando, van a ver una vista desde el robot que está encima del otro y ven el cable con que se conectan, así que hay una vista completa desde el primer robot hasta el fondo. Van a estar bajando a 4.000 metros de profundidad después que terminen este transecto a 1.200 metros y están en lo que se conoce como la cara este del cañón de la Mona.
Thank you, Graciela. We're getting closer to 1,200 meters for our last midwater transect.
Depth 1195, slowing. Okay, that's 1,200.
Okay, we're at 1,200 meters.
Bringing my tilt.
Ready to start our last transect.
All right, starting at the top of the hour.
Okay, we're starting 11 a.m., 1,500 UTC.
This is our last chance, man.
It's our last shot.
Tilt.
Copy that. Pushing out. Tether, switch sides.
No, I was thinking, based on the view that we get from the wide eyes, the HDs for the manipulator volume, just wondering if the
Okay, we're about 24 miles northwest of Puerto Rico.
HD
Conducting our last midwater transect of the series on the 5th dive of the Oceano Profundo expedition. We are going to then head to a bottom depth of 4,000 meters.
The HD bubble up.
would do better on Sirius.
And survey the east wall of Mona Canyon.
Something just went off screen to the upper right.
Upper right.
Upper right.
You see it, Andrea?
I don't.
Yeah, I've lost.
Okay, we can continue.
Sorry, Mike. Something. I'm gonna go in on that video. Video zoom. Oh. Hey, Brandon.
Okay, we're going to zoom in on this object in the center screen.
We're stable.
That's looking like a low-bait ctenophore.
Just hold there. And
wow.
I don't think your mic is on video. I can, thank you.
Another really important gelatinous group we haven't seen very much of on these midwater transects.
Just hold there. Get it more into our lights. There you go.
Yeah, that's great. We're getting a view kind of right from under the lobe. Now it's coming around.
Sorry, just hold there. I'll come back. Thank you.
Ctenophores are like are gelatinous, but they're different than the medusae that we see here. They don't have stinging cells.
Give it a little more, Brennan.
Ctenophores
If you've got it.
typically use sticky cells, or a lot of these low bait ones use mucus layers to trap prey.
Just hold there.
And they're characterized by teen rows, or these comb rows, for propulsion and for some assistance capturing prey.
Pretty impossible to focus on, huh?
Yeah, and the classic invertebrate zoology text, they talk about ctenophores swimming with the cilia in their comb rows, but these big lobates in the deep sea swim with a frog kick.
Did you say a frog kick?
I think that's what I said.
That was a good description. Do you guys want to try to get a better zoom on this, or?
I think this one's called a blood belly jelly.
I'm trying to figure out why I'm oscillating so
The Bathosauroi.
Come on, Via? Yeah, thank you.
A blood belly?
Blood belly. There's a little red spot of red right in the middle of it.
Yep, I agree with that.
Keep coming if you got it. Try to fill frame with it. Hold there.
No, yep, can see it now. Very well. You can see the cilia moving and reflecting that light back.
A couple more seconds on this, Jared, sorry.
A lot of times people will mistake that for bioluminescence when what they're actually saying is reflection and refraction of the lights of the submarine.
Okay. Let's let him go. Come wide. Thank you.
Thanks, guys. That was great.
Hey, Dave. I just have a question.
Copy. Thank you, Brennan.
Wow, that was really cool.
Go ahead. Does our auto depth have the bandwidth to take out the heaves?
Negative.
No.
It's got 1 meter resolution.
And
1 meter? 1 meter resolution? Oh, wow.
Really?
I don't know whether it's truncated or rounded, but that's...
honestly, I think that was
There
me.
have been some recent
Doing a
papers
lot
that
of
have
possible,
suggested that
thought it was
ctenophores
pretty
have
well
actually
timed with the
evolved
heave of the
before
Sirius.
the peripera, before the sponges.
Just the football floats.
Yes, some of it was.
We can go into data view.
Which morphologically
that
doesn't
over.
really make a lot of sense, but I think more evidence comes out that that may be
That
true.
was me, though.
I know it's a little contentious still, but
Oh, you were looking at football, the footballs, and
Status.
I think we have 2 more minutes in our 1,200 meter transect.
Maybe try the NRV nav solution.
This is rather annoying. Oh, here. Oh. Looks like we have 2 decimals of precision, actually.
Okay, a depth 1,200 meters. Been conducting midwater transects. Michael Vecchione from the NOAA National Systematics Laboratory and Michael Ford from the National Marine Fisheries Service have been here with us leading these midwater transect objectives, and they've been doing this now for the past couple years, using the Okeanos as a tool to look at the midwater communities.
it's right there. Little jelly. Come in video. Very small.
So that concludes our 1,200 meter transect.
How much more you got, Jared?
So watch lead, that's the
Plenty.
end of
It's a baby.
Okay, thank you.
Come a little wide.
Thanks, Nav. So that was the end of our last midwater
There it
transect.
is.
You want to zoom as it gets to the center? Yeah.
It's a tiny jelly.
He is small. Okay, video. Come on.
Full wide.
Okay, so it's the end, Nav.
That's the end.
Well, thanks, everyone.
Yeah, thank you, pilots and video. That was amazing. Great images. That squid in the beginning was pretty amazing. Sorry you guys missed it.
Have to remind us.
And thank you to Mike Vecchione and Michael Ford.
Backing up here a little.
So push on down.
Thanks, that was fantastic.
Go
Another
to
successful
tow,
set of
continue to
transects
descend.
there. Really great.
Yeah, and if you come port, I believe that'll...
I could do this all day.
Yeah.
Well, I'm
Yeah, I know we got in the water a little later today, but I
facing
think
the
it
way I
was
need to
still
be
very
facing.
important to conduct these midwater transects before we rush to the
Oh,
bottom.
I see. Okay, sorry.
There's a lot we do not know about this
descend and
pelagic
go out of
environment.
auto-head, you'll get spun around. Well, actually, you should turn yourself around and stretch out so you don't get snatched.
So what's the next?
I'm going to go ahead and sign off now.
Just
All right, thank you,
continuing
Mike
to come
V.
ahead. Okay.
Talk to you later.
Okay, talk to you soon.
Delta's 28, so you're
copy that.
You're safe.
All right.
What's the estimated time to the bottom?
I am turning.
Stand by. Nav,
Starboard.
estimated time to bottom.
Got about an hour. We have about an hour and a half
And
from
pushing.
now.
You're tugging me big time. Just
Yeah.
calm down.
So it'll be 11:40 local. Sorry, 12:40 local.
12:40 local?
Yep.
Oh, okay, good. It's earlier than I thought. Graciela, do you want to do any Spanish translations?
Sure.
Oh, we have a little bit of echo.
Pegaron los de la columna. Should I stop?
Nope, I think you're good now.
Okay. Ya se terminaron los transectos de la columna y a 2 personas con esfuerzo de la NOAA y pues mayormente lo que vieron fue una serie de medusas, sinóforos que también vieron algo de mucosa cuando ya los organismos no están en ellos, exoesqueletos de crustáceos y estaban alrededor de 10 minutos. Así es que tuvieron una discusión sobre cuánto se veía aquí versus cuánto se había visto en otras áreas. Empezaron la buceada un poco más tarde de lo previsto, pero de todas maneras se llevó a cabo porque ahora hasta las 2:40, que es cuando llega el robot al fondo a los 4.000 metros de profundidad, van a estar previstos. Pero de todas maneras se llevó a cabo porque ahora hasta las 2:40, que es cuando llega el robot al fondo a los 4.000 metros de profundidad, van a estar bajando por la camarones con unas antenas muy extendidas. Yo me pedí el calamar, espero que ustedes lo hayan visto. Y pero todas estas cosas van a estar disponibles en el internet como parte de los videos que se dejan para que el público los pueda seguir viendo. Toda la información que se está recopilando, todas las fotografías y videos que se están haciendo, que son fotografías del video pero de alta resolución, van a estar disponibles primero para los científicos para que puedan identificar las especies que se vieron, y como habrán oído a través de la narración de ellos, podían identificar muchas de especies, hasta géneros, sino hasta especies. También se estuvo discutiendo sobre las diferencias entre estos organismos y describían ellos cómo era cada uno de estos organismos. Yo no estuve durante toda la mañana, así es que espero, y por lo que oí, sí hicieron muy buen trabajo haciendo las descripciones básicas
Yes, that is
de,
correct. I have
de
about
estos organismos y la
245
información
meters
que hay es que la
Okay,
columna de agua
would
es
it
el
be possible
mayor
to extend that
espacio que
move
hay
so that
en la
we
Tierra
are
y es de lo que conocemos
285
más poco,
meters
así
from
como
our current
conocemos
position?
también muy poco del
Same
fondo del mar.
thing,
Y
73
comparativamente, aunque
degrees.
tenemos un mapa completo de
No problem.
Marte,
I
por
will
ejemplo, de
make
la
our
Luna,
final
pues
destination
no tenemos esa
285
misma
meters from here
información
at
del
073 at
del
210
océano, que apenas 5%
Thank you,
o
Nav.
así del total del océano
Okay, Dave.
se
Got
conoce.
you in tether.
Autohead
Y
is
con
off.
el 70% de la Tierra es agua en vez de tierra.
All right.
Así que ahora ellos van a estar esperando a que lleguen al fondo, que será alrededor de las 12:40 del mediodía, y entonces estaremos de
So
vuelta.
perhaps we will just
Este,
continue on this
esta
course.
investigación se conoce como
45
Océano
minutes.
Profundo y es del
About
Okeanos
88
Explorer, el
minutes to
barco
the
de
bottom, so
exploración
29
de la NOAA,
swing under.
que está ahora mismo a 24 millas al noroeste de Puerto Rico
I'll let you
y
catch up a little
estará buceando
Okay, I'm bringing the
de
speed up.
4.000 metros hacia arriba durante la tarde.
Bump.
Así que muchas gracias por la atención de todos y espero que sigan acompañando especialmente a Andrea y a Mike, que son los que están en el barco haciendo todo el trabajo. El resto de nosotros estamos en tierra mirando desde nuestras computadoras y comunicándonos a través de teleconferencia con el barco. Así que los dejo hasta esta tarde. Andrea, I'm going to go
Yeah, thank you.
do some other work, and
Okay.
I'll be back around 12:30 or so to catch up with you again.
Great, thank
Is that
you.
okay?
Yes, absolutely. I'm glad you can join us again.
And thank you very much for the great job that everyone's doing.
Yeah.
It's been wonderful.
Yeah, thank you.
Gracias. Bye.
Bye-bye.
Push out a little more.
Okay, and we're headed down to a bottom depth of 4,000 meters. We're going to be surveying the east wall of Mona Canyon. We're in pretty much the same general location as we were yesterday, where we surveyed the west wall of Mona Canyon, just about 12 miles to the west.
So, Brian, to answer your question about depth servo, Josh just reminded me it wasn't that it was 1 unit of depth or 1 meter depth resolution. It was a 1 second update rate. Is the sticking point.
1 second update rate.
I do believe.
1 hertz.
And the other issue is thruster deadband.
Yep. Yep, understand. Yeah, I remember that one. Like I said, I was just curious.
Just give us something to work on while we're sitting here while Chris does all the work.
Yes.
Happy to.
Speaking of doing work, Chris, would you like me to stow your light bars? What's your pleasure?
I would. In
All
fact, we can redeploy once we get down there. No squid, Brian.
Oh, no.
Pretty
Halfway
bummed.
between sad and disappointed.
Yeah. I'm all the way both.
Okay, okay, good, thank you. So, Chris, are you free for a couple minutes to tell us a story?
That depends on the story.
Keep in mind your EXRV is...
Yeah. Are you talking to Pilot Chris or...
Yes, Pilot Chris. No, I'm just curious. I think we've all had different paths to education and just life experience-wise to get where we are today. And I was just curious if you could tell the audience, what kind of your background is, how you ended up in the pilot chair.
Sure, I'd be happy to. So I went to school at Virginia Tech. Go Hokies. I majored in aerospace engineering and ocean engineering, double major.
Wow.
And coming out of school, Coming out of school, I was very interested, and I read a couple of articles about similar work, deep water robotics work in Yellowstone National Park.
Oh, really?
And
wait, in Yellowstone?
In Yellowstone Lake.
Okay.
And from there I sent some emails out, some cold emails to people that I didn't know, and offered to volunteer my services, said that I was very interested, was wondering how I could get involved, and a few emails later I heard back, and in the meantime I had accepted a position as a naval architect for the Navy.
Oh, wow.
And through a mutual understanding and some cooperation, I wound up here. So, yeah, I've always been interested in the deep sea, working on the water, and here I am.
And how long have you been with the program now?
Oh, since 2011.
Okay.
So 4 years.
Wow.
Or 3.5. I think this is my 6th cruise, I believe. But
what was your first cruise? Was it Indonesia?
No, it was a little bit after that.
Okay.
It was Panama Canal to Key West, so it was a Cayman Trench cruise. So I met the ship in Panama, and then met a lot of these guys, and have stayed entrenched ever since. Luckily,
no pun intended.
No pun intended. I'm not that clever.
Thank you.
But yeah, now I wound up here, and I don't intend to leave. I enjoy it very much.
So you're one of the people that likes their jobs.
I am. Very much.
So it begs the question, how did you initially get interested? Just something popped in your head and you said, I wonder if anybody's doing ROV work in Yellowstone Lake.
No, I've spent a lot of time out there in Montana. I think I was reading an article, and I'd seen some specials as a high schooler, middle schooler on kind of ROV work, some shipwreck work, and just kind of exploration in general. And I've always been really interested in that, kind of looking at things that nobody's ever looked at before, and that was just sort of a gateway that I saw as a possible opportunity, and sent out a million emails, got one response, and here I am.
So was that what led to the dual major in ocean engineering and space? Well, not aerospace. Was it aero?
Aerospace and ocean.
Aerospace, yeah.
Yeah.
So go up or down, one of the two.
Yeah. Both work with fluids. It wasn't all that much more work to get the double, since it was something like 12 more classes or something. Since they have a lot of the same core concepts between the fluids, air, and water. So
just working across density boundaries.
Yep.
Just different Reynolds numbers.
Yeah, exactly.
Where'd you grow up?
Baltimore, Maryland.
Okay.
get involved in any robotics projects at Virginia Tech, they have quite a few people. They have this is Dave Smallwood.
The name rings a bell. I can't put a face to it. To him,
fleets of AUVs, and
yeah,
there's a guy in mechanical engineering who does like robotic mechanisms. They did the DARPA Grand Challenge. But can't remember his name now. But Virginia Tech got like second in the urban challenge. That may have been when you were still in school. I don't know.
Yeah, it must have been a mistake. I'm sure we deserve first. Now, I was involved with the human-powered submarine for a bit.
Oh, okay.
Which
yeah.
is, but yeah, they've got all kinds of stuff. Like you said, a fleet.
You're not going to put in a plug for Ritter Designs? No. That would be using federal dollars.
Yeah, that's right. It's,
okay, yeah.
And we've just crossed a depth of 1530 meters, 1530 meters.
Is that your first Midwater transect?
No, I don't believe so.
Okay. First is cruise, though.
Yes, and first one feeling this comfortable with the new vehicle.
Yeah.
After working out the tweaks, my personal tweaks in piloting.
Your personal tweaks. Nav watch lead.
Go ahead, watch lead.
How long will we have on bottom? When will we have to leave?
We'll have approximately 3 hours on the bottom.
Okay.
Depends on how high we get before, the intersection of those 2 points. But I estimated we'd come off the bottom around 3,500 meters, and that would leave us our time to go around 4:35.
4:35. Okay.
Yeah.
All right. Thank you.
I think I'm trying to get rid of this flare. I think it's a reflection off the craft, to be honest. On my bottom left, you see it?
I feel like we've had that.
Oh, the
flare.
craft is not stowed. It's leaning out. I'll bring it up and see if that kills it.
I feel like we've had that flare longer than we've had the craft.
You've got the hydraulics running. So.
On the bottom left, that big
yeah,
flare?
but you know, but moving, checking it out isn't going to hurt anything. So
Hydraulics are already on.
Yeah, I'm going to turn them up a little.
Okay.
All right, all your cams are wide too, so you get the best field of vision for this.
Way down. Oh, saggy, baggy, saggy.
Turn it. Oh, that's actually, yeah, I was thinking turn it the other way. It doesn't change the flare. It certainly seems like it is related to the flare.
Yeah, it is certainly changing the flare.
Yeah.
It's not going away, but it's...
Get out the black spray paint.
There you go.
There
now
you
I've
go.
got the craft out of the light,
Yeah.
facing way out wide to that side, and it's gone.
Sure enough.
So if you bring the craft in and then tip the starboard lower swing arm out a little bit, would that do the same thing?
Well,
I don't remember seeing that flare quite as bad at the bottom.
It's out at the moment. It's all the way right, I think.
No, the swing arm is, tip it outwards.
Oh.
Deploy it a little bit.
Deploy it a little. Yeah, that might do the same thing.
Let's try it. If you bring that in, and I'll take the starboard lower and tip it out. I'll wait till you're all the way in. Big bright light shining.
Hold on, can you push the bubble cam down a little for me so I can see that distance? Oh, I got room.
Yeah.
Actually, leave it right where it is. Maybe if I bring it all the way in here, it'll do the same thing on its own.
This is a good view. Your mini Zeus.
It's about as far in as I'm comfortable going. What do you think?
That's fine. Yeah, so I'm going to deploy the lower swing arm a little bit and watch it.
Yep. Why weren't we... We didn't have this before.
There, it just went away and I
Yeah.
barely
Well,
tipped it
yeah,
out
that
past
looks
the
good.
arm.
Okay, great.
So let's leave that.
Sounds good.
Hydraulics off.
Todd, are the lasers off?
Lasers are off.
Okay.
Here, 12K ohm with the craft on.
Yeah, okay.
It's pretty
It's
horrid.
pretty bad.
Yeah, so you have to go hunting.
In the arm itself? Yeah, start unplugging
We
connectors, see
can
what happens.
start with the back of the shell and figure out what... Right, all the pots share a ground and a power, and then have their own returns. Transducers likewise.
So
And
just
the valves likewise. Well, they each have their own signal. They all share common ground.
So put a bender between each one of those pins.
Well, I would start with the multimeter at that level.
Yeah, that's true. Depending on
Hey everybody, this is Mike
how much
Ford. I'm going to
capacitance
close
is
down
in
the
there.
Silver Spring ETC.
Okay, thanks,
Yeah.
Mike. Thank you
ROV,
for joining
I'm
us
looking at some
today.
of the tools that you have in the bin in front of the arm. Can
Great,
you
thanks
talk
a
a
lot
little
for
bit about
the
those? Maybe
Midwater
tell us what those are.
transects, and I'll check in toward the end of the
Yeah.
dive.
Okay, great. Have a
They're
great
not
day.
so much tools as practice blocks. They're just aluminum blocks. Since we have the craft and we plan to use it, some of us younger guys who haven't played with it need to practice grabbing different types of handles and practice reaching over and grabbing something, pulling it in and out of a bio box or a punch core or any kind of things like that. So,
I see a T-bar, a ball, and some rope. Are these standard handles that would be used for?
yeah, those are the standards. There might be other options, but those are the three most common. So, I guess the rope is, since it floats and it's a nice little loop, it might be amongst the easier. The ball's probably the easiest, and the T handle's probably the hardest to manage. So we hope to be sampling and possibly using different tools that have those kind of handles, so getting used to using the manipulator. So that's their only purpose.
Thanks.
I guess if I tilt down on the Mini Zeus here, you can actually see a couple other things with balls on them, and they are actual tools. Right there on the front of the drawer, below, right, you can barely see it past the Zeus Plus. That's our temp probe that was permanently mounted to one of our manipulators last year. We've put a ball on it, so now we can grab it with the craft and still use it as a temperature probe. And then you can sort of see, can you tilt all the way down and see it if we deploy the drawer?
Yeah, and we're actually sending
there
out
it is.
the
Look
Zeus
at that.
too. Oh, wow, that's great.
That's our tool. So that's the temp probe, and it's on a set of magnets there. And the cable runs through a nice blue it's similar to our yellow hose, but it's got a coil already in it.
Okay.
And then we have a cable attached to that. It goes back to a reel. So as you grab it with the arm and reach out, that cable will all deploy. And then when you go to put it away, it's self-managing. It just finds its way back into that. So Todd designed that whole process. And then you can just see the ball and the magnet on a plate off to the left side of the Zeus Plus, and you can see it. That's our sediment poker that we were using last year.
The famous sea poke.
The famous sea poke. Yeah. So those are our actual tools that we have aboard. Those are things we're hoping to use, and we still hope to use once we manage the craft.
Great. And for the benefit of our viewers, I also noticed, Todd, we have a sliding tray.
We do.
And that will be used for
the tray for tools like you just saw,
Mm-hmm.
the temperature probe and other future type of samplers or tools or samples. And we're going to add on to the front of it, onto either side of the vehicle, the two lobes that you see where some of these practice samplers are mounted. They're going to have.
We've just passed 2,000 meters, 2000.
So it seems like the design philosophy of the ROV is to try and keep a lot of the tools modular, so you can add and subtract and change as the mission also does that.
Yes, and the other thing that is unique to this vehicle is we've tried to keep the front view of the main HD camera very clear.
Mm-hmm.
Put the Zeus Plus, the main HD camera, on D2 as low as possible. So when we nestle in next to some organism or critter or coral or whatever, we can get as close of a view as possible. Get really great macro shots. And we didn't want a big front-end loader type of ROV where you have stuff in your face all the time obstructing the view. So the way we're going to be designing the drawer appendages, that it'll stay out of the way, but we'll be able to articulate them inwards. So our arms can reach them in the center of the vehicle when we want, or put them out of the way. Keep our view clear.
So this vehicle is very, very custom and purpose-built for exploration and research as opposed to a repurposed industrial ROV.
I think that's true. The team here on board, we're the ones that have really taken this vehicle from the frame up, built it for this purpose.
Thank you guys very much. I appreciate the explanation.
Another flare to chase.
That was there. I noticed that all day yesterday.
Yeah.
I'm not sure where that's from. What's bright and shiny? I wonder if it's the that little nickel plate that we added. I wonder if we should try to black that out.
You think so?
Look down with your mini Zeus or bubble can and have a look. I think it's pretty bright and shiny.
Yeah, it is pretty bright and shiny down there.
See that?
Yeah. So if I brought my dome all the way up, that would disappear if it was that, right?
Your dome all the way. Yeah, it should. Although then you're going to be looking up towards your lights and you have a whole nother
Well,
set.
there it goes. See? It's disappearing.
Okay.
Yeah.
What else could it be?
So right now I can't see it, and that seems fairly in line. It could also be the temp probe or any of like the nuts or any of the stuff on the sampling stuff out there.
I wonder if it's the,
Well, here, if I
not.
got an idea. If it's on that side, it's most likely coming from the port, right? Do you agree?
Yeah, I think so, yeah.
Port lower,
Jim,
it's
what do
sort
you
of
think?
opposite.
We're looking at
usually,
the,
right?
there's a very, very small flare in the HD image on the top right.
Yep.
Trying to figure out what that might be coming from.
The left toe.
Yeah, so something
what we
over
thought
there,
too.
yeah.
We thought maybe the seat pokes mount, that little piece of nickel for the magnetic mount.
Well, I think it may be just. I mean, generally that's pretty light out there. It doesn't have to be a specific thing, I don't think. But
That is awfully bright and reflective, though.
yeah, it's true. You could play with turning off some lights and see if you. Yeah, that got rid of it.
Yeah.
Which light was that?
That's just the port main.
Port main, yeah.
Yeah,
the ones
so
that point
the
pretty
one
much
that
down.
aim at all that stuff, yeah.
And it went away, for sure.
Now you can see other things. I think flares keep popping up as you get rid of other ones.
That might already have been there.
Yeah.
Masked.
Huh.
We do have a black plate that we could cover the craft mount with. All of this stuff can be.
I don't think
dark enough.
that's. I mean, I don't think
It's
that's
doing
as big a problem as things out on the toes.
You're probably right.
I mean, it's nice when you're looking with the mini Zeus. It kind of can start blinding you, but
Yeah.
with this camera, it seems fine.
Those tools that you've got on the toes are mainly aluminum, correct?
Yeah, they're aluminum
The
blocks
boxes are
painted.
plastic, but yeah.
Okay. Could you maybe have those anodized?
Yeah, they're
Just so
painted,
that they
dull gray right now.
Oh, are they?
Yeah.
We have
All
a
right.
professional painter. Very carefully prep and paint those for us. You can see the blocks themselves aren't very reflective at all compared to the
nut,
fastener
yeah.
on the top of that ball.
True.
Yeah.
They are significantly hotter than the rest of your front porch area, though.
They are.
Yeah, look at that. That whole thing just kind of glows. Well, note to self, make the bio boxes black. Whatever you put out there, make them gray.
Yeah, well, most of that stuff is black.
Cover them in
basically, right?
felt.
I mean, black or dark gray.
I don't know how long that would last,
All
though.
the stuff out there?
Yeah.
Front? Yeah, black or dark gray.
You wouldn't know it
Yeah.
looking at it right now.
It's true. Do you mind restarting this?
Yes.
The mouse on the.
Sorry,
right
you
there.
pointed to that earlier.
Yeah, you were talking. I just.
It's this guy, right?
Yeah.
Or is it CNET V2?
It's this one. They're the same now. This is the old version 2.0, and when I downloaded CNET Pro on this, it went out, found the old version, updated it, and it downloaded another version. So, or it deleted the old version, I think.
Pick up to 33. Looks like you're getting a little bit ahead of me.
I just backed off a little, just a touch.
That's max.
Okay.
That's all this thing. That's full throw.
If you can go to 30, then I'll just.
That's 32.
Yeah, Chris has got a joy lock on and then just a small Z bias.
Yeah. It's got a lot of axial thrust on it, given the state of our port side.
Yeah, but
back that off.
I was wanting to back it off, but as soon as I do, then my joy lock will kill my verts too.
Oh.
So I'll have to compensate and then play with the axial, which is why I was commenting the Z bias.
Whatever you want. Just crank down on the Z bias.
Yeah, it's true. All right, be ready for some changing. I'm gonna go ahead and just fix this real quick.
Yeah, no problem.
Okay, that's where he had it. Just gonna try to turn up Z bias while I let off on the thrusters and try to keep it close to the same. Okay, so I had 20 something Z bias and 65, so somewhere around there.
Yeah, you were like at 69 or 70%.
Yeah, 69 or 70 plus 20 Z bias, so you think a little more?
No, what you see here is what you get. So you should back it off because you're already getting way beyond me.
Oh, I see what you're saying. Yeah, okay. That's true. Alright, back in action. I can actually back off a little if you want to catch up some.
Yeah, I think you better push out a little bit.
Pushed out some.
Or, no, never mind. My bubble cam on Sirius has had a pretty heavy zoom on it. So I got a little nervous there for a second. Yeah, you're okay. Dave left this with a big zoom on the Sirius's bubble cam, and I saw you getting pretty large in that image.
Gotcha. Okay, I'm gonna ease off on this a little. Let you catch up some. Just gonna hover at 45 for a little bit. 45%. Let you catch up. Maybe. Do you see me?
Okeanos, this is UNH.
Hello, this is Okeanos Explorer.
Hi, it's Mimi at UNH. I was just looking at the senior in the order column, and I thought I'd say, oh, I have some UNH scientists here.
Hi, Mimi. How are you?
Doing very well. So lovely here in New Hampshire. Everything has finally arrived.
Good. Mimi, can you hear me better now?
Where is today's dive?
Today's dive is on the east wall of the Mona.
Yes, I can hear you very well.
Good.
So today's dive is on the east wall of the Mona Canyon. We're diving to 4,000 meters, and we're attempting to try and look at the canyon wall to look at both the biology and the geology. And
The bridge has calmed down a lot.
we're attempting to get below this big, thick platform carbonate sequence of rocks that we saw yesterday. The whole northern side of Puerto Rico and the coastal margin is covered by this big sequence of carbonate rocks, which are from anything from 1 kilometer to, in some places, 2 kilometers thick. And several of the dives we've done so far on this leg have been looking at that sequence. And today we're going to start at 4,000 meters and try to get beneath it, see the rocks beneath it, and then come up slow. And as always, we'll be looking at the biology, and we'll also be looking at the possibility of landslide evidence, because this is a very deep canyon. The canyon where we're diving goes down to 5,000 meters. That's 5 kilometers below the sea surface. And we'll be down there between 4 kilometers and 3 kilometers, partly trying to understand how the canyon formed.
I keep turning right to try to like fix it, but it doesn't seem to.
I'm just gonna let it go the way it wants to go.
This is like a dive that's done before in other dives, and this will be the deepest V dive in that area.
Seems like you guys are in line with each other.
We've actually done 3 dives to about this depth, at about 4 kilometers. Our deepest dive will come at the end of the cruise. We're going to go to the north wall of the Puerto Rico Trench and look at something completely different. We're going to look at oceanic crust and not continental crust. And that dive will be down to the full depth range of D2. We'll go down to 6 kilometers.
What's that? Yeah. I guess just more forward.
Well, that's the squeeze system. Well, I hope you guys have a great dive today, and we'll definitely be checking in.
Thank you, Mimi.
Thank you, Mimi.
Thank you.
Bye.
What's our bottom target? 4,000 even?
Yeah, 4,100 or something.
Minutes. I dropped the thruster.
Is your thruster even running?
It wasn't. It was a minute ago when I was twisting.
So good afternoon, everybody. It's noon on the 14th of April, and I'm broadcasting from the Okeanos Explorer. We're lowering the D2 ROV to start a dive on the east wall of the Mona Canyon. Our target depths today will be 4,000 meters, coming up to about 3,500 meters, and we're currently passing about 2,500 meters. So we'll be on the sea floor in about 40 minutes' time and starting the dive at about 1:45. Our target for today is to try to look at both the geology and the biology. And in particular, we're trying to get beneath the big platform carbonate sequence, which is about 1.5 kilometers thick, but varies in thickness. And we're hoping by going down to 4 kilometers below the seafloor, we can see beneath this big carbonate sequence. Carbonate sequence formed from about 30 million years ago to about 3 million years ago, and it covers the whole northern coast of Puerto Rico and offshore towards the Puerto Rican Trench. Below it is the core rocks of what Puerto Rico is made of, much older island arc rocks. Puerto Rico was once very much like Japan today. It was a volcanic island arc formed off the coast of South America, the western coast of South America, and it's those old rocks that form the basement on which the carbonate rocks were looked at. We'll also be looking at slope failure structures, inevitably diving on these steep walls, and this wall is 3 kilometers high, which is an enormous wall, really. We'll be looking at how the slope fails.
Coming up on 5 meters
and seeing if we can see any evidence for slope collapse, slumping,
Thank you, Bridge. Sure.
etc. And of course, we'll be looking at the biology, which so far on this cruise has been spectacular, and hoping to see some strange and exotic animals and record what lives down at those depths. So again, we're hoping for another successful dive, and we'll be starting in about 40 minutes. Co-pilot video. So
yeah, go ahead, Ed.
I've set the input error setting for the transom wire cam to bypass.
Okay.
So it should pass through video that it detects as having an input error.
Okay.
Same change we made to the AFPTZ. So
I wonder, you know, amongst the possible errors, I wonder if it's gamut, because when it goes to bypass, it sure looks over-chromed.
No, I think
no, I'm seeing that. But we don't see that
this
on the
could be a potentially tricky
monitor
landing
itself when we
not
look
really
at
a
the
landing spot.
composite
Roger that.
analog.
Potential for current.
Understood.
Try to estimate the slope.
Oh, yeah. The camera's still pointed way out there. I think from our transects.
Yeah.
Not that that's a issue.
I'm in the center of the light pool. I have the best opportunity to catch something interesting. There goes a larvation.
So what's our target here?
ETB, so we'll be done in about 40 minutes.
Okay.
Our estimated time on the bottom. We'll zoom out. So we have an ascent of this wall, pretty big wall. Slope at the beginning is around 30 degrees. Slope in here is, we'll feel like 90 probably.
Yeah.
And we want to go, I think it's about 1.3 kilometers if we have time. Nope.
Oh, wow.
900 meters, sorry.
What is going on there?
I think that's just
a shelf, or is that an aberration?
I think that's
Might be
in the con. Oh.
Might be.
Sorry, I got tracking on.
Might just be an anomaly in the contour.
I think it's just in the contour drawing, yeah. It's just they connected 2 points here straight across.
Ah, understood.
Missing
Seems interesting.
pixels. Something somebody edited out because they didn't think it was real.
Okay.
But the backscatter was hard returns up this slope, so they're hoping to see hard substrate, maybe corals, although it seems pretty deep.
Okay.
That's all I know.
So it's a 600 meter-ish ascent from landing to top of
Yes.
subject. All right.
Brian Nav, watch lead 2 here. Yeah, if it helps. Very similar to yesterday. We don't quite know what we're going to find, but basically we want to go upslope from a geology perspective, looking for an outcrop. We might be below that carbonate sequence that we saw yesterday, and in which case, if we could actually find the boundary between the stuff below and the carbonate, that would be great. But, similar to what we were doing yesterday, stopping to look if we see rock in situ, looking at the loose stuff a little bit until we know what it is. In truth, we could be on a talus slope. I mean, it might be like yesterday, all carbonate again that's fallen down the hill. We just don't know. And then as for the biology, it'll be the same as yesterday, stopping when we see cool things and trying to get great video.
Yep, got it, watch lead.
Roger that, Mike.
So Ed, video, the Pilot Cam SD is the composite output of the...
Yeah, it's a third one in, correct?
Yes.
So I decided to take a look at some of the settings on the signals we are not having issues with.
Okay.
Amanda, Hui, can you hear me?
There's a lot of video proc settings on that one. That was the first one I looked at. Let me look at another.
And of course, that analog version of it is just a backup.
Right.
Of the
HD
on fiber.
Yes.
Yep. Okay.
I just wanted to see what some of the settings were compared to the ones we're having issues with. Minor changes on the ports LPS. They're all wiping out the vertical banking interval, though. They're not passing it through. Look at one of our problem inputs.
So pilots, while we're all together, I have started a bit of a punch list. I'd like to install the chafing gear on the tether. I'd like to spend some time with the craft. There was discussion of swapping the port axial motor.
Yeah.
I'm looking at Dave.
I flushed it after the first dive, made quite a mess. After the second dive, it didn't look so bad. I don't know how it looks now.
I flushed it twice since then with Carl's help last night.
Okay, so it might need some attention.
Yeah, it's the shallow dive. It was fine, but it's spitting a lot of stuff into the comp and gas, unidentified gas into the lines.
Okay,
so maybe after recovery today, we'll
assess.
We have one
And
pretty much
then-
ready to go, right?
I don't know.
I think I was told that we have one ready,
I think those guys are on it down
but
there.
has not been wet. So,
Okay.
but
What else?
tomorrow's dive is short, not short, shallow.
Okay.
Is that end of list?
There was discussion of, and this is hypothetical discussion at this point, but there was discussion of moving an instrument from the ship's CTD rosette onto the Sirius CTD. I believe it's an optical backscatter type of instrument.
Transmissometer.
Yeah, maybe LSP or something like that.
Yeah, if it's
It's very preliminary, and it was a request, and I think
Right.
it's working That its must way be up the the, chain.
yeah,
ST8 they were talking about yesterday.
it's. I guess step one is the ship has to agree to loan it to us.
Right, and then,
And
I mean,
then
I think we
sort
can
of-
evaluate whether or not it's technically
Yeah.
feasible, and then, you know, they need to decide the ship and the protocol for integrating instruments on the system,
Right.
et cetera, et cetera. But
Right. There's the question of is it plug compatible with the 9
Right.
Plus,
Yeah.
and then there's the calibration.
Yep.
And configuration.
like I said, just hypothetical at this point.
Okay.
But that's on my list.
All right.
Anything else that we should have on the list?
It's much lower priority, but we have not resolved the question of why the new camera did not behave properly on D2.
I've had some
Which camera?
discussions about that. Oh, the
Yeah.
new HD?
Yeah.
and I have some things we could try if we want to. I don't know if we can bench mount it and
We
connect
can
it to the
bench
CCU.
mount it.
Yeah, I have
Wow.
some things we can try.
Yeah, I put it on the board down there to bench test. That's something that can be done during a dive if the guys have energy.
Okay.
They need to go through that procedure anyway and get familiar with all the cables and stuff.
Right.
That's been built over the years.
Yeah, I feel like we don't have. I mean, it's a little tougher to get tasks like that done during the dive with a longer day.
Yeah.
You know, I mean, I personally, I feel like I need that off time. You know.
Yeah. I'm just.
Because by the time we all do launch, we all do recovery, and we all do 5 hours.
Yeah.
It makes for a full day.
Right.
You do a little more than that.
Yeah. And then with taxes, which is my current dilemma.
This is also not time critical, but maybe a little winch of would be good down there. I think there's some grease piling up down there. Dave had the idea of ordering some sort of removable sheet that we can.
I think we put a liner in there of some sort, held in with magnets.
Good afternoon, everybody. We have just passed 3,000 meters.
Carried out
As
instead of rolling
D2
around.
dives down once again on its fifth dive of the cruise, down to 4,000 meters in the Mona Canyon.
That need to be
We are aiming to be on bottom in about 30 minutes' time. So the dive observations will commence in about 30 minutes.
Part of the lore.
Running out in front of you there a little.
All right.
Just a little touch up here and there would be good if. I mean.
Just shovel out the big patties.
Yeah. I mean, I could do it one of these days too, mornings or something.
So it looks like Sirius is settled out. Might make a small move to try to hit the spot. I don't think they're terribly interested in the spot, but just while we have another 1,000 meters.
Yeah, Brian, you know, basically we just want to go up slope.
Yeah, understood. Watch lead. So we'll just stay here.
The telepresence guru is is looking at the thermometer and is going to report in.
Reporting in on our very accurate, I'm sorry.
Yeah.
We are currently at 80 degrees.
We should increase cooling a smidge.
The engineers have the calibration chart for that if you need its certification.
Okay.
Good. I just want to make sure that everything is up to ISO standards.
Yeah, ISO 9002.
From our Bed Bath & Beyond thermometer. Occasionally, mapping will close it overnight. I'll check right now to make sure it's open.
And so the other recommendation from the engineers was, first keep the curtain closed, but if that doesn't work, to close the door.
Ah.
Oh. Sounds like we have more flow.
Vent deployed.
Thank you, Biddy.
So, having gone down that list, the
Time
highest
to put the fleece
priorities
on.
are motor swap and craft.
And I think the tether chafing is something I would like to.
Yeah.
And the rest are as we have time.
Yeah. Remind me,
I must. This is a problem.
D2 passing 3,300 meters, descending.
Nav, the winch making about 33 meters a minute now.
Sorry, come again?
Winch is running 33 a minute now.
Okay. This is Andrea Quattrini on the Okeanos Explorer. We're just crossing a depth of 3,365 meters, 3,365, headed to a target depth of approximately 4,000 meters, surveying the east wall of Mona Canyon today. We are about 12 miles northeast of the site that we surveyed yesterday, the west wall of Mona Canyon. Today we're on the east wall of this canyon. And we'll be on the bottom until about 16:30 today.
Chris, is that motor that's ready to go already have a cable on it and everything?
I do not know. It's a, I would assume that Bobby and Jeff have that. They're usually pretty good about having stuff ready to go. So, okay. And if they don't, I'm sure they would be happy to get it up and running.
Just thinking about task division. If it's already got a cable on it, then that frees Bobby and Jeff to work on the craft in the control room, which I think is the next logical step, is to replace the cable in here and then go into our testing sequence.
Sure. And if we decide to do that tonight, I can pull the prop and all that.
I don't think that'll happen tonight. I think we'd come up early tomorrow, give ourselves a little maintenance time.
Copy that.
But that has not been authorized yet. So.
Understood. You can see that artifact still kind of hanging around in there, Dave, on the starboard manip cam that I sent you on a wild goose chase after.
The little thing at the top there?
Yeah, it's worse depending on swing arm location and craft orientation. But I've noticed it a lot since I told you to run chase it.
You looking at the two little white dots?
Yeah, there was occasionally there's a larger, more solid reflection in the middle. And on the first dive, I told Dave to go look at it. For some reason, we couldn't find it. Getting out ahead of you there a little.
It's exciting.
We're at 3,500 meters.
Yeah.
3,500.
And about 15 minutes to go, and we'll be on the seafloor.
Did your ears pop? Well, what was the highest drum temperature for the shallow dive?
I think it got up to 100-ish, didn't it? At one point, Carl was telling you linearly increasing or something.
Well, that was the extrapolation. The question is, did we reach it or did we level off? I haven't looked at the log yet.
I think it was up towards that. I know yesterday when I was checking, it was 93-ish.
That's pretty typical.
Yeah. So on a shallow, and that was deep, so on a shallow dive, I guess it'd be 100 isn't out of the question. When do we worry?
I disagree. I think you're doing a fine job. Mike.
Well, the specification on the cable is 150 Fahrenheit.
Okay.
That's when the polyethylene starts to soften and its mechanical properties get a little scary, and then it starts to move around, then the electrical properties go to pieces.
But we're not reading temperature on the cable, we're reading temperature on the drum, so it could be higher on the innermost cable.
This is absolutely correct. I pulled a number out of the air, saying that 120 would be a cause for grave concern. There's no thermal model to back that up.
The thermo is a suspect.
There was a model done, and but it modeled the wraps, the layers, as a solid chunk of steel. So it's not to be trusted. Now, did you see the work?
So this is Mike Cheadle from the Okeanos Explorer. We're about 15 minutes away from reaching the seafloor. Today,
I remember you doing it.
Today
What did you do?
We've been taking bets on what we actually might find, and the three possibilities are: we might find a talus slope,
basically,
we might find in situ carbonate, or hopefully, the most exciting of all, we might find the rocks below the big platform carbonates. And if we do, we'll traverse upwards and try and find the contact with the carbonates that lie above. Overall objectives today are to look at the geology, to explore the fabulous biology we've been seeing, and to look at the slope morphology, looking for evidence of slope failure, landslides, trying to understand the nature of the landslide hazard around Puerto Rico.
Measuring it at the drum.
Yes.
Hopefully it'll be an exciting
and also
dive.
at bit of the wire that was exposed to air.
Yeah.
Just because that's the only other place I could get to it.
I guess.
What was ambient?
70 something.
But to get a gradient sort of radially from the drum to the outer edge, like you'd want a temperature difference between the inner drum and the outer, and then you could estimate the resistance in the radial direction and know kind of what what gradient it would support.
Yeah.
So if you shoved some bags of ice inside the drum and measure the temperature outside and in.
I think what I wanted to do is just get it under the top wrap.
Mm-hmm. Yeah.
To isolate it from the radiative and convective effects, and then it's an easy test to repeat.
Yep.
There was also some question about getting the optical, the fiber optic measurement instrument on a short-term basis.
OTDR or something?
It's not an OTDR. It uses some optical property that has a thermal
thermal coefficient to it, yeah.
And it gives you, it both uses the TDR principle to determine the location.
Way out front, yeah.
So it'll give you a temperature profile along the whole 8 kilometers.
Oh, wow. They give you a profile of temperature as a function of length.
Yes.
Oh,
that's what's advertised.
Wow.
That'd be extremely useful.
So
then you know everything.
Then you know everything. Originally, that was proposed as a full-time, real-time for the system, but now we realize that if we can just back out a model for the gradient and resistance, then that drum sensor would be a reliable proxy.
Exactly.
That doesn't give you temperature at points. It would just give you an equation for the entire length for the temperature on of the wire, right? So you could back out everything else you needed.
Mm-hmm.
Yeah.
Well, the fiber measurement it gives you both distance along the fiber and the temperature at that distance. Yes. That's what's advertised performance now. Resolution, accuracy, et cetera, et cetera, et cetera.
Did we just cross into a layer with more particulates?
Seems like it, Andrea.
And we're at a depth of 3,729 meters.
But so far, I've never seen the interior temperature climb above 100.
Yeah.
How reliable are these temperatures, Dave?
Those are pretty good. And that is measured internal to the transistors that are switching, turning DC into AC at variable frequency. So that's, you'll see that go up.
So we're getting closer to our bottom target.
Very quickly.
About
the heat sink.
4,000 meters depth.
Not so good.
We are surveying today the east wall of Mona Canyon. We're about 30 miles northwest of Puerto Rico.
They just seem to be about a degree and a half different, and they haven't been working for a while. I'm just curious.
And it might be worth just outlining what Mona Canyon is. It really is an enormous canyon. Where we're diving, it's about 30 kilometers wide. It's about 50 kilometers long, but its extension ends, it carries on much further than that, and it's about 3 kilometers deep.
And we're really in an area where the Mona Canyon is part of the Mona Passage, right? So we have water coming in from the Atlantic that is coming in through the canyon and through the passage into the Caribbean Sea. So this is one of the conduits of water exchange from the Atlantic to the Caribbean, and then also from the Caribbean back out into the Atlantic.
That's correct, and it's one of the reasons why we have to be pretty careful, because you can get some pretty powerful currents coming through that passage, and currents are bad news for the ROV, and so we have to monitor the currents very carefully.
And so some of the water masses that are coming through, mostly in the upper layer above about 300, 400 meters, include Caribbean surface water, subtropical underwater, and Sargasso seawater. So the subtropical underwater and Sargasso seawater are really formed in other locations within the Atlantic, and they are moved through the passage into the Caribbean. And then under this, there's tropical central water that exits through the passage back into the Atlantic. So really interesting area in terms of oceanography and geology.
Indeed, and I guess the big interest of the geology is that we only understand it in the most simplistic way. The canyon itself is some sort of rift created by literally stretching and pulling apart this part of the continent, pulling away Puerto Rico from Hispaniola, but we don't really understand why. And that's one of the objectives of these dives, to try and learn more about how Puerto Rico and its surrounding areas react geologically.
Video pilot.
Go pilot.
Hey, are we clear on a white balance, or would you like another one down here?
I would like to do another one.
Copy that. You will have to talk to co-pilot.
The co-pilot's getting his ducks in a row.
We might, certainly won't complete it by the time we get to bottom, Dave. So
I'll just stop a little short here.
And this is an interesting area as well, especially for shallow water organisms. There's been studies on
pilots are with images indicating the bottom of the
reef,
series
shallow water reef corals that
4030,
show different
4,030
populations on
meters.
either side of the Mona Passage. So this area can serve to be a genetic break. Oh, we have an announcement.
Okay, Chris, refresh my memory. We want...
so this area with these currents moving through the Mona Passage can really serve as an isolation mechanism and causing populations to become isolated on either side of this area.
okay, you have port on. What's your RPM?
RPM.
HPU stat. Yeah, there you go. Oh, that's plenty.
Yeah.
Yeah, you should be good then.
Just passing
elbow up.
3,900 meters, 100 meters from the seafloor.
So that's good. But
video watch lead.
Elbow up all the way. Clockwise, all the way. On wrist. And shoulder yaw in. And,
this way or the other?
That way. Sorry, counterclockwise. I told you wrong. And then you might have to elevation up on your shoulder and kind of forearm, wrist play until you get it square. And then, of course, you'll need a swing arm action to light it up.
Okay,
but
let's see.
I'll let you do that. I'm going to keep an eye on depth here.
Yeah, do that. Let me get some light on the subject. Let's see. Upper swing arm. Do you believe I need some down?
Andrea, am I too loud now?
Okay.
And your right lower
Better.
is slightly deployed.
Okay.
To eliminate
we'll see how this goes.
I want to hear everything you're saying. Absolutely.
I know you're just trying to tune me out.
Pilots, do you mind if I push
No,
out
no,
the
no.
Sirius sonar and see if we can
Go
pick
right
it
ahead.
up?
Negative. I think.
So yeah, to give you all a perspective, we're sitting here in the ROV control room with microphones on. We're listening to those shoreside participants that are calling into the telecom line, where we can listen to all the pilots in the front, a videographer. Even if we would want to, we could listen to what the bridge is saying, and the deck crew as well.
Keep coming.
And we can also talk to all those people as well through these microphones.
Sure.
Indeed we can.
Stand by.
And I think sometimes they
Some
hear us
combination
when
of
they don't want to.
shoulder
Probably,
elevation and
probably.
elbow will get you square. But it looks somewhat good.
Tilt down. That's you, Chris.
Oh, sorry.
So Andrea, what's your hunch about what we're going to find down here?
Stand by.
I have a hunch we are going to see a lot of
So-
cladorized sponges as we
Should
did
I be
yesterday.
concerned at all that we're
Possibly some predatory
Getting some
tunicates we
altitude
saw
hits?
yesterday as
good?
well.
Okay.
30 meters from the bottom.
Hoping
Yeah,
we see
Dave, let's
more
come to a stop
corals.
here.
And more fish?
Probably not more
Which all
fish.
stop.
I think we'll see the same fish species and the
There we
same
go.
abundances as we saw on the west wall. I would be surprised if that was different. But we are exploring an area that's hopefully not as covered with soft sediment. So maybe there'll be more substrates for corals to successfully settle and colonize and grow into adulthood. So hopefully we'll find more corals and more coral
Nav, I'm
species.
gonna hang here.
Okay.
Okay.
Watch our delta.
So we
Yeah,
are
now that we have it in
30 kilometers
sight, so to
away
speak.
on the other side of the
Just-
canyon.
Or
Can
we'd
we
be more closer
say anything about whether
430.
we'd expect the fauna to be different, or can we
Do you have
tell?
any idea
It's-
what the
I mean, I don't think so. I think if the
Backscatter
micro
was?
habitats were the same, on either side, if
For
we
the
saw
wall?
the same type
Yes, it should be hard
of
substrate.
substrate,
That's
I think we
one of
would
the
expect to
selection
see the same type
process.
of fauna. But with
for
the
this is that
substrate
it-
being
well, it's pretty
different
bright. I
on
mean,
this
it's
side,
30 degrees
if
though, so
there's less sediment cover,
for-
then we may
gotcha.
see
For the
more
ADCP,
colonization abundance.
for the
I
DBL,
would
you're getting some
suspect
beams
that
that are hitting it
species should
pretty
be the
far
same.
down.
But,
understood? Yeah.
on either side. Now, if we get shallower, we may see different species. So in the deep sea, a lot of community changes we see are
Auto
really
depth
zoned by
and
depth,
auto
or
heading
by changes
on.
with water masses even.
I feel
Thank
good
you.
about my
Yeah.
position.
Yes,
I don't
yes.
know. We can zoom some more.
White
video.
balancing.
Did you rotate that lower?
I did.
So has Amanda Demopoulos and Jason
left one might
Chater
help,
joined
too.
the Hui ECC?
Did right and left.
Can we bring
Yes, they are
the
here.
camera up
Hi,
or the arm
how's
down?
everyone there?
Go ahead and take that, Dave.
We're doing great. It's Tim Taylor.
I'm gonna keep
Just
an
so
eye
we're
on
centered on
Amanda.
this.
And Taylor Heil. Hello, Taylor.
Stand by. I'm gonna slow it down.
We're at max zoom right now.
Good afternoon, all.
What's that?
Hello, hello.
Could try
We're
to
ready.
pump it left, but I might overshoot.
Good. Just keep those typing fingers ready.
Nice.
Perfect.
They are ready.
I don't know about my planarity. Looks a little bit. There's a little focus shift across there, but should be good enough for a balance.
Well, let's give it a shot and see how it looks.
Okay. If I'm still low on light, I might be able to make some adjustments, but
Gotta tug you back home.
Yeah, I'm not really happy with this. We're still
Okay.
pretty low on light.
stand by. I will work on this.
I'm gonna wide out a little bit so
Yeah,
you can
yeah, I can see what's
get a better
what's
idea of what's going on
Let's
exactly.
see. Starboard upper, swing arm,
No.
down.
One of the uppers is
Ah,
off.
ah.
There you go.
That's what it was.
Better.
Try that.
Much better.
We also have a suite of shoreside scientists participating with us in the chat room. While I have the chance before we get to the bottom, I just want to thank, I'm going to go down the list, Amy Bacco-Taylor, Chris Kelly, Eric Cordes, Christopher Moore, Lindy Nelson, Liz Shea, Mike Cheadle, well that's you,
That's me.
Randy Singer, Santiago Herrera, Scott France. Thank you guys.
And Amanda.
Great. Amanda, Jason, Tim, and Taylor
White
at
balance
Hui.
complete.
And Graciela is still on the phone. Is she still on the phone with us?
That's my fault. Tugging.
See how that looks when I pull it back a bit. Those vectors look pretty good. All right.
We clear?
Clear and happy. Thank you much.
You're welcome. We put things away. Let's see. Upper swing arm, light up. Lower swing arm. Starboard, we want to go right.
And then also shout out to the folks on the coordination side that have been helping with this coordination. Brian Kennedy, Brendan Reeser, Catalina Martinez, Casey Cantwell, and many others. Thank you guys.
Take my camera back.
Andrea, this is Jared at Video. I took watch lead 2 down a couple dB and let me know if that sounds more even.
Okay, thank you. Audio check, Mike.
Hi, this is Mike Cheadle on the Okeanos Explorer, about to start dive 5 of a series of 20 dives to the bottom of the seafloor around Puerto Rico.
The yaw is sticky.
Yeah, we need to re-engineer this.
I think we just need
Video
to
watch lead, it's
adjust
better
that
than yesterday, so I can work with that. I'll be fine.
Okay.
It's an issue.
We should stop.
Get down 1 more dB for good measure.
Fix it with the electronics.
If you, yeah, I would.
See, I believe this goes that away.
Sure does.
Oh, we don't want to go all the way around though. Where do we stow this? There or
you can figure it out better if you yaw all the way left and then shoulder all the way up.
Okay.
And it's usually chip chart to the port side.
Amanda was just suggesting I should go next door, and then I'd be less loud.
Oh no, already started. He doesn't even know what he did.
Maybe you need a name tag, Andrea.
I think so.
Wrist back, just
branded across my forehead.
I couldn't resist.
This thing's backwards. I know we've had discussions about that.
You want to yaw all the way left too?
Thought I was yawing all the way left.
I still got some there.
Hi, Casey. I just saw that question. We got a question from one of our listeners. What effect would there be on your dive if there was an earthquake while you're exploring? Well, if we're in the water, it won't affect us really, except if it caused the slope to fail. In which case, if the ROV was on near the slope and suddenly that the earthquake shook the slope enough such that it started to lose rocks, we'd try and get out of there as soon as possible. But that's really quite unlikely. The real big earthquakes to cause that sort of landslide are quite rare, maybe happening once every 100 to 200 years. So we'd be pretty unlucky to run into one of those. And the small earthquakes that occur all the time, really we can't really feel them, as people, perhaps yourself, living on Puerto Rico, don't really feel the small earthquakes that are going on all the time.
Right, all right, that looks good.